Yeah, hi everybody. My name is Matteo. I'm a postdoc in Jack Gallant's lab, and I'm so excited about today's workshop because after seeing all these great talks this morning, I was like, I want to go down to the scanner and get some new data to do some really cool stuff. So this project, we're interested in looking at the anterior temporal lobes and doing high resolution mapping of the ATL. And the reason is because the ATL is very critical for conceptual knowledge. It's a very critical part that shows up in patients, and it is altered by many different conditions, many different clinical conditions, but also through aging. There is aging targets this area. However, as you probably know, this area is really hard to image. So with Joseph Wu, we started doing some sequence optimization at 3T using, first of all, this approach that we use in the Gallant labs. This approach pioneered by Alex Huth in 2016, where basically we have people listening to narrative stories while they are in the scanner. And then we fit what we call voxel-wise encoding models to essentially try to predict their brain activity from the semantic content of the stories. And by evaluating them and interpreting them, we can then recover semantic maps or maps of what different brain areas are representing. And as I was saying, we started this ATL project at 3T with Joseph Wu, and we developed this ATL optimized sequence. So here I'm showing prediction accuracy of the semantic models. And as you can see, the comparison, actually, let me just use the laser pointer. We do get considerable improvement in prediction accuracy when we compare this ATL optimized sequence, which is a multi-echo sequence, to a standard sequence. And using this sequence, we are able to recover very rich semantic maps in the anterior temporal lobes. And so since this looked so promising at 3T, we were like, okay, let's do the same at 7T. Not only that, today we saw so many cool results using diffusion. And so we want to put the two together. We want to be able to recover the semantic maps at high resolution, hopefully matching the same resolution that we get with the TI, to try to do this multimodal mapping of the ATL with the goal long term to try to understand how the ATL changes with aging and in these various disorders. So I feel like this part is basically done. We need to figure out exactly how to optimize a sequence for naturalistic imaging at the 7T. And so, with naturalistic imaging, like listening to stories, there are different constraints. Like we cannot really have multiple repeats of something like stories because the signal that we measure will change. So we need to increase SNR for this type of naturalistic imaging. And the way that we typically try to gauge if a sequence actually gives us signal that is useful for naturalistic imaging is using this quantity called explainable variance. So instead of looking at the sequences through the lens of temporal SNR, we're going to use this metric called explainable variance. And what I'm going to show is essentially this type of stimulus. It's just a 2.5-minute long movie, which we repeat like 5 times. And by repeating the stimulus, we're able, I'm going to stop it because otherwise it's too confusing. We're able essentially to measure the repeatability of the signal. And because our voxel-wise encoding models can only for now predict signal that is repeatable, we really want to boost up this explainable variance to be able to use them for naturalistic stimuli. And so just to give you an example of what it looks like at 3T. So this is a flat map. I don't have time to talk too much about it, but essentially this is occipital cortex. This is frontal areas, medial and ventral areas. But as you can see, this stimulus activates, or we can recover high explainable variance in the ventrotemporal lobe, in early visual areas, in auditory areas. You will see that it doesn't activate a lot of DTL. I'm going to touch on that later. But essentially, we are going to be using this stimulus as a first take on selecting some sequences that may work at 7T. So we, thanks to Alex, Erica, we collected some preliminary sequences and which I consider as a baseline. These are like whole brain And in the interest of time, I'm just going to focus on this 1.16 millimeter whole brain sequence with a 1.75 TR. That's another constraint that we have because we have naturalistic stimuli, so we want to have a TR that is fast enough to be able to model this data, this naturalistic data. And so when we look at AVSNR, sorry, at expandable variance with this sequence, again, this is repeating the stimulus 5 times. So this is about 12 minutes worth of data. Here I'm showing only the expandable variance for the middle echo. These were all multi-echo sequences. And when we use the combined echoes, we do see that there's an improvement in functional SNR, kind of reiterating what we had found at 3T. We can look at the actual percentage improvement. And one question is this just because of averaging, or is it because we're actually sampling some functional signal that is TE dependent? And if we are now binning the voxels based on essentially the optimal T2 star estimated from the data, we find actually, let's say this is in blue, we see voxels that have an optimal T2 star towards the middle echo. We actually find high EV for the first echo versus the later echoes, and we find this pattern consistent for the other echoes, showing that it's not just averaging, it is there is an advantage of averaging, but we're also getting better sampling. So yeah, this is just the beginning. I'm really excited after seeing this morning presentations what we can do more for this. And with that, I want to acknowledge people at UC Berkeley, UCSF. This project is also funded by the Well Neuro Hub, and so we have collaborators at UW. Are really welcome. Thank you.